Hello all, welcome you to Medicine 2020. So today's topic of discussion in brief about is the clotting cascade. We'll discuss as simply as possible and in a very quick and non-forgettable manner. Okay, unforgettable manner. So this is the clotting cascade that we are going to discuss about. So there are basically two pathways, multiple factors involved in the clotting cascade, which activate each other in a form of sequential events. So that is why it's called cascade. A cascade essentially means a sequence of events, one triggered by the previous event. Okay, one leading to another. So these clotting factors activate each other in a fashion that in a step ladder fashion, we can say probably, and including some other ions or other factors. So basically, what are these clotting factors? If we discuss them, what are these clotting factors? Clotting factors are zymogens. What are zymogens? Zymogens are, they are pre, or you can say precursors of enzymes. Okay, or inactive form of enzymes. These are converted into serine proteases. What are serine proteases? These are the enzymes which break the peptide bond. Okay. So initially these are the things that we need to know, the two definitions, basic definitions. And following this, we will know we have a word called activation. Okay, what is activation? So activation is nothing but conversion of an inactive form of a factor into active form. So how it is converted, what are the things that an inactive form has and active form doesn't, or what makes it active. So to know that, we have to know one thing. There are few changes that occur when the clotting factor is synthesized from the DNA through a protein synthesis. After translation, there are few parts in the clotting factor. For suppose this is inactive form. There are few parts which make it pre or pro form of the clotting factor. This end part contains some amino acids which makes it inactive or pre or pro form. They are removed in the post-translational modifications, which makes it an intermediate product kind of thing, which contains multiple amino acids, and this contains two parts. One is binding site and one is active site, which are non-functional yet. Okay, which are non-functional yet. So there is this middle part holding these two together. This middle part, this form, these amino acids are cleaved. So okay, this protein, this entire protein that we see here is being cleaved by something, right? This entire protein is being cleaved by some enzymes which are present in the blood. Usually these are those which activate or initiate the clotting cascade. Okay, we will discuss soon. This middle part is removed by, again, proteolysis. As this proteolysis is not breaking entire protein, and only it is breaking a small part, or this proteolysis is or breaking down the protein in a limited manner. We call it limited proteolysis. Okay, limited proteolysis. So this, after removal, this active form contains two sites. One is binding site. Okay, another is activation. This activation is nothing but enzymatic. This enzymatic site, that means it processes a property of enzymatic action. So these two are linked by disulfide bond. So this factor now is the active form. Okay, it has a binding part and an enzymatic part. This activated form, with the help of this enzymatic part, will again do a limited proteolysis of the next factor. Okay, as we have multiple factors and two pathways which initiate the clotting cascade. Okay, so in an easy way to remember this, there are two pathways, right? Two pathways. So what starts these two pathways? These all clotting factors are written in Roman numericals. That means they are 1, 2, 3, etc. Right? So these two pathways are started by Roman numericals containing 2. Okay, 2 is written in Roman like this. Right? The bigger pathway is intrinsic pathway. That means it is started by 12. The smaller pathway is extrinsic pathway, that is started by 7. Simple. Now, if we go further, these two paths will cross at a certain point, right? These two paths will cross at certain point, and this is 10. Okay, cross at 10. This is also over. Now, what are the things that activate these initiation factors? This 12 is activated by high molecular weight kininogen, kallikrein, and collagen. Okay, when this factor 12 comes in contact with any of this, it gets activated. That means it is limitedly proteolyzed. And after that, this 7 is activated by tissue factor. Right? It is present in the tissues whenever there is an injury, it is released. It is nothing but factor 3. Okay. So after that, if you are doing a project, okay, this is a work of art. The clotting is a work of art, and these clotting factors are the individual artists. And if you are doing some big project like clotting the blood, you have to check with someone who is very elder to you. Okay, you will show it for corrections and all. And the final factor in the cascade is 13. This is fibrin stabilizing factor. Okay. Now, if you take this from 12, if you go to 10, if you descend one by one, 12, 11 is there. Sorry, 11 is there. Is converted into 11A. This converts 9 into 9A, because this already, there is already 10 here, so there will be no 10 after 11. 9A. Here this 9A is converting 10 into 10A, and this 7A is converting 10 into 10A from the extrinsic pathway. This 10A is converting prothrombin into thrombin. This thrombin is converting fibrinogen into fibrin. This is added upon by factor 13 to cause stable clot. This is the simplest way you can remember. Okay, done. Here what is happening? We are left with two more factors, factor 5 and factor 8. Right, factor 5 and factor 8. So here, where is 5 adding? If you take this as 10, what is half of it? 5. In a Roman, half of it horizontal, half. So here it is acting along with 10A to convert prothrombin into thrombin, and 8 is left. 8 is acting along with 9A, its friend. 8A is acting along with 9A to convert 10 into 10A. This conversion of 5A, 5 into 5A and 8 into 8A is multiplied by thrombin. Okay, so that means thrombin itself is increasing its own activity by converting 10A. Okay, increasing the 10A. Now this is the clotting cascade. It's not tough to remember. It is very easy, right? So after that, what is happening here? Where is calcium used? The calcium is used in few places. That is, one is 11A along with 11A, 9A, right? 10A and 13A to stabilize the clot. These are the places where calcium is used. Also in 7A. Also in 7A. This is the extrinsic part. Okay, 7A. Here is the calcium used. And if you take one more thing which is very interesting here, we have few factors called vitamin K dependent clotting factors. What are they? They are 2, 7, 9 and 10. So why are they vitamin K dependent? What is happening? So let us see. This vitamin K is making them into something useful by gamma carboxylation. That means it is adding carbon dioxide to it. Carbon dioxide. This addition will provide a site to bind calcium. This addition of or this process of gamma carboxylation will provide a site within the molecular structure for calcium to attach. That is why gamma carboxylation is important, without which the factors are useless. Simple. Okay. And coming to all these problems where the diseases that we talk about, there are multiple clotting and bleeding disorders which occur here with this. With the problem in clotting factors, they are hemophilia, 3 types, A, B, C, right? And vitamin K deficiency will cause which factors? Problem with which factors? They are 2, 7, 9 and 10. 2, 7, 9, 10. And here the hemophilia is A is caused by factor 8 deficiency. Hemophilia B is caused by factor 9 deficiency. And hemophilia C is caused by factor 11 deficiency. Right? And whatever these factors are there, they are vitamin K dependent. Vitamin K is synthesized and stored in liver. If there is a problem with the liver, these clotting factors will be defective. And in the newborn also, these clotting factors are not yet produced or activated because of lack of vitamin K. That is why we give them vitamin K in newborn. Vitamin K in newborn. So simple. These are the problems which are caused within the clotting factor. Also, there is one more important point. This thrombin which is present here. This thrombin facilitates the conversion of 13 into 13 gene. See how much the thrombin is acting. Okay, so this is in brief and in detail about the clotting cascade. I hope you understood and it is of much use